Good morning, everybody. Today, my presentation is about the assessment of soil micronutrient setting in northern Ghana applying gamma array spectroscopy. In sub-Saharan Africa, food insecurity, including feeding hunger, is a very key issue. As a result, up to 37% of the world's stunted children are said to live in sub-Saharan Africa. This, among other things, may be attributed to micronutrient deficiencies. Unfortunately, there is no consistent data that maps systematically the flow of micronutrients from the rocks into the soils, into the plants, and then into the animals and humans eventually. Also, information on the spatial distribution of soil micronutrients is not usually available in sub-Saharan Africa. So in this study, as a first step towards assessing the micronutrient chain, we developed a site-specific and efficient method for micronutrient chain mapping using gamma ray spectroscopy, which is what I'm going to present today. Now, this is basically the approach that we use in terms of the work that we've done so far. Here, we talked about the fact that we, along a transect, road transect within the target area, we took up to 100 samples, soil and rock samples. Now, these soil and rock samples were analyzed to determine the mineralogy, the total nutrient content, and the plant available nutrient content. But these are basically point data. And our goal is to transfer this point data into the entire region. It's more like an extrapolation right now. So the next step is to also acquire the airborne gamma data, remote sense, for the entire area, which is basically what you see here. So the goal is develop a model that links this point data, soil data to the gamma, and then you can now extrapolate. Nevertheless, of course, I'll talk about that issue that arose somewhere along the line, which is what you see here, and which that's the fact that we wanted to go back to the field and then get point sensing of the gamma spectroscopy for each of those points where we collected soil samples to kind of generate the model first, making the points, and then extrapolate to the larger area. Now, in terms of the results we've acquired so far, if you check for iron and manganese, there's no problem within the study here because all the areas have adequate amount of iron and manganese. For copper, 61% of the sites have high concentration. For zinc, which is where we get the problem, 74% has low, which is inadequate. For molybdenum, 95% of the area has low, which is inadequate still. So the goal is these two are problematic. This one to an extent, this relatively adequate. But when we go to the next level, which is basically now moving from the point to the spatial area. Now, here we try to develop more like for the first step, a linear correlation, simple linear correlation between total potassium content in the soil and the remote sense potassium. You can see that there's a good correlation, but then for thorium and uranium, it's relatively weak. So we look at the next level, which is that we'll go back to the field and get a proximal sensing, taking moisture content into consideration. And of course, we'll also do a multilinear correlation using covariates like soil moisture, organic carbon, clay, and pH to improve the mapping process. Subsequently, if I were to conclude, because I think my time is almost up, I'll say that plant available macronutrients in the topsoil is variable throughout the study area. There's potential for effective mapping of the spatial distribution using gamma spectroscopy, but the process needs to be improved, which is what we're working on right now. And finally, the next step of this study is actually now talk about how the nutrients flows from the rocks into the soils, into the plants, into the animals, and eventually to humans. Thank you.